Thank you very much. So for those that don't know, Labcorp, Covance, formerly known as Covance, is now the drug development arm of Labcorp. We were acquired back in 2015, but rebranded more recently under the Labcorp name. So after just a brief introduction, I'll talk about some of the preclinical models and approaches for looking at CAR T-cell therapies, which can be utilized in a variety of other cell therapies in the oncology space, and then talk about some of the biomarker approaches in this area. So as we all know, this is an area that's been actively growing and developing over the last few years. And in May of this year, almost 1,800 products in the development, some stage of development, with about 1,300 industry-sponsored cell or gene therapy clinical trials going on globally. And this pipeline really helps feed the FDA projections for 10 to 20 new approvals per year by 2025. And we're seeing, of course, astronomical amounts of funding being poured into the industry, which continues to spur the development of these types of products. So when we look at the development of cell therapies, a whole host of in vitro or ex vivo assays are required in order to fully understand and characterize them and screen them for things like transduction efficiencies, viability, vector copy number, cytotoxicity, et cetera, and then be able to track their overall persistence and cell killing capabilities once you move to the in vivo setting. And within oncology itself, there are a whole host of models that continue to be useful in the cell therapy space, and I'll just briefly touch on some of those. So when we look at the human cell line models, there are hundreds, if not thousands, of these models commercially available. And they've been a gold standard in oncology development for a long time, but they're also quite useful in the cell therapy space for a number of reasons. Many of them express the targets that are of interest, and there are a whole host of hematologic lines commercially available. Of course, they've been around for a long time, so there can be genetic drift, and we find that from one lab to another, the characteristics can sometimes be different, and so understanding that baseline performance of the model will be important depending on the types of questions that need to be addressed. And of course, though, you need to run these in immunodeficient animals, and so that can be somewhat different from the actual patient setting, of course. And then the advent of patient-derived xenograph models provides another whole host of model types that are out there. They're more extensively characterized, but it does require generally more precision and ability to develop these types of models and keep them established over long periods of time. But also an immune-deficient animal is required. And in the hematologic malignancy space, the generation of these PDX models can be very challenging and can also be quite limited. So again, understanding the particular performance of particular models and the questions that need to be addressed will be important in choosing and selecting the right model. And then to get around some of the immune deficiencies, the humanized approaches are, of course, out there, and they continue to go through evolutions of changes for expressions of cytokines or other areas to help improve, say, myeloid engraftment or other approaches. And so we're starting to see more and more requests to talk to clients about particular humanization approaches and how they can be utilized in the cell therapy space. And of course, as we start to see and hear more and more over the last few years about the movement away from hematologic malignancies and the holy grail of cell therapy of really being able to have an impact on solid tumors, there are a whole host of these human cell lines that can be utilized in the solid tumor setting, either in the subcutaneous space or orthotopically implanted into the region of interest. But we tend to still see large numbers of hematologic malignancy studies being requested, and so utilizing in vivo bioluminescence imaging in order to track disease progression and therapeutic benefit. You can utilize models that we've heard a lot about at the conference already, CD19-driven models or multiple myeloma or AML models, in order to evaluate things as shown here with this experiment where we looked at a CD19 positive model. You can stage by the in vivo imaging, treat with a CAR T, and then evaluate over time things like 2 different types of CARs to screen them in vivo, or even look at a dose response over time. And you can take serial blood draws in order to look at persistence of the CAR in those animals during the duration of the study. And so the continued use and development and enhancements of in vivo models helps overcome some of the challenges and future trends that we see. And I don't have time to go through all of these, but another trend I'd like to focus on is the continued use of biomarkers and their role in cell therapies. So we all know that this is a highly complex area, and that because these therapies modulate the immune system, it's important to not only think about what we know and how we characterize the product itself, but also the tumor and the tumor microenvironment of the individual patients that are being treated. And so when we look at a variety of biomarker applications in cell therapy, we can look at markers that evaluate expansion and persistence of the product, but also functional and phenotypic characterization of that product, establishing pharmacokinetics, pharmacodynamics, and mechanism of action for efficacy and how that ties together. But then, of course, how do we use these markers to further be able to predict response, to be able to look at safety, and even relapse or resistance to these types of treatments, ultimately to be able to use many of these biomarkers in order to identify patients and stratify them appropriately into clinical trials. So once a cell therapy is delivered, in order to look at that pharmacokinetics, you need to evaluate the cellular expression and persistence of the target, and you can utilize things like flow cytometry or PCR methods in order to evaluate persistence. But also, because these are immune modulatory agents, they can be immunogenic, and so looking at immunogenicity to the product is important, as that also can have a direct impact on the overall efficacy of the therapeutic. Another key area for biomarkers and looking and thinking about them from a strategy perspective is to look at biomarkers that can affect safety or can potentially predict safety of a cell therapy. And many of these biomarkers can be soluble factors like ferritin or C-reactive protein, but also a whole host of cytokines that can be overproduced and result in cytokine release syndrome. And we know that many patients following treatment with a CAR T product can have cytokine release syndrome. And being able to have actionable biomarkers in order to potentially predict those patients, so to be able to monitor and provide therapeutic treatment with things like tocilizumab or other therapies that can help would be very important. And so being able to do those immunoassays and identify those patients, we're seeing more and more of that these days. And of course, it would be important to be able to look at markers that can help predict or stratify patients by the biomarkers that are out there. And this gets us into looking at, biomarkers that, what can we do, because the therapy it's an immune-directed therapeutic, so it can have an impact on the immune system. So you can look for biomarkers of against things that would impact the immune system, so looking at immunosuppressive markers, looking at things like TIM3, LAG3, PD-L1, et cetera, or even looking at markers that can identify memory response or looking at memory phenotypes of the T cells. But also, as I mentioned earlier, these types of products, it's important to also look at what it does to the tumor and to the tumor microenvironment. And this is where we start to see the utilization of transcriptomics and being able to identify through more broad screening profiles, looking at RNA-seq or nanostring or other applications like that, where you can start to try and determine what biomarkers are causing response or non-response to particular therapies. And then there are a host of surrogate biomarkers that one could imagine utilizing, such as tumor mutational burden or other possibilities along those lines. And so a paper that came out recently from the Moffitt Cancer Institute, they asked up front whether or not a patient had a priori factors within their own body to be able to differentiate between responders and non-responders following treatment with Axicell. And so they identified or did biopsies from these patients that had large B-cell lymphoma prior to treatment with Axicell. And they did a variety of profiling of the tumor, and the image on the left is the expression profile of patients that had durable response on the left side of that, and then on the right side, non-durable response. And they found a whole host of interferon gamma signaling genes that were upregulated in patients without a durable response. And then they asked whether or not those patients that had a durable response or non-durable response also had an impact on Axicell expansion. And the volcano plot shown on the right then looks at the interferon gamma signaling genes in the yellow squares correlated with patients that had poorer expansion of Axicell, so making the correlation that prior to treatment even, that those patients had underlying upregulation of these interferon signaling proteins that correlated with poor expansion of Axicell. And so it really, I think, is a nice example that shows how these biomarkers can be used in a potentially predictive manner to be able to then stratify patients that you think may or may not respond as well to your cell therapy. And so we know that this is a complicated field, that there are no one-size-fits-all models or approaches, and so it's important to consider what's the right model for the right question that's being addressed, and then what particular biomarkers and what's that strategy to help identify and characterize the product, the safety of that product as you move into early clinical development especially, and then are there biomarkers that can help identify the right patients to put onto the right trials for the right drug. And maybe I have 2 minutes and 30 seconds left, so I think I talked a little bit faster than everybody else, but happy to address any questions now or speak to anybody later on that wants to know more about how we can help develop your products with you. So thank you very much.